Thank you for bringing me over to Boston again. If it's only virtual, I will. I was there for a couple of years as a postdoc, and I really enjoyed my time. I think we did some science that still influences how I think about certain things. We are doing certain things differently here than we used to do them in Boston, which is also good. So I will give you a little bit of an idea of what we've been up to in recent years and what the future might bring. So first of all, I went for a slightly simpler title than the one I had, single-cell biology, causality at scale, and cell therapy. And I will try to give a talk without too much epigenetics because I see my epigenetics research slowly aging, and I think we need a big paper on epigenetics again anytime soon, so I can bring that back. But today it's an almost epigenetics-free talk. I'm in Vienna, and I should kind of extend my regards from Vienna. It's a very nice place. If you haven't been here previously, visit us at some point. If you like like me, you like the glass flower collection at the Harvard Museum for Natural History, you will definitely like the 18th century Vox models of human anatomy that we have in the Josephinum Museum of Medical History here. And kind of where, like I work, I'm just sitting here, just a little bit up here where there was vineyards in the 18th century and kind of one of the largest hospital complexes of any European city built next door. And wherever you walk here, it's historical ground. And in many ways, we find inspiration from the science that happened in the 19th century in Vienna. I want to give two examples here. The work of the Human Cell Atlas to which I'm contributing can be seen as a 21st century kind of revamp of what Karl von Rokitansky did in the 19th century, looking at thousands and thousands and thousands of autopsies and tried to correlate what he saw under the microscope in terms of sick tissues, diseased tissues, to the phenotype of those patients. Kind of for the first time, really connecting cell tissue phenotypes to patient diseases. And Karl Landsteiner, one generation later, discovered the blood groups and for the first time enabled cell therapies that were safe based on kind of a precision medicine understanding of what that not the blood of different people follows certain rules. And I will give a little bit of an example how we try to use some of our technologies towards cell therapies and programmed cells as therapeutics. But let me kick off with an example of why I'm so excited about the combination of single cell and spatial technologies and the work that the Human Cell Atlas is doing. Like Okitansky, who wanted to understand what a disease is like. This question is still something that we face on a day-to-day basis working with our clinical collaborators, because for many of the rarer diseases, the more heterogeneous diseases, we do not understand very well what is going on. And in those cases, the combination of single cell and spatial profiling is really the first step towards understanding of an understudied disease. And I want to give an example here, sarcoidosis. Sarcoidosis is granuloma formation for unknown or no good cause. Granulomas you might know from tuberculosis. These are these kind of immune cell compartments that essentially shield the pathogen away from the body to stop an immune response that is no longer helpful, even though the pathogen cannot be fully eradicated from the body. And in sarcoidosis, this happens for kind of bad reasons. And one example is here is tattoo-associated sarcoidosis. A tattoo works so that the color particles are injected really deep into the skin, and there they get phagocytosed by macrophages. And they sit there essentially for a lifetime deep in the skin inside macrophages that shield them away from the body. Problem only is that those tissue-resident macrophages have a life expectancy of maybe 2-3 years. Eventually, those macrophages break apart, and other macrophages have to come in to again take up these particles. So you could argue that a tattoo that someone might get in their teenage years will stays for a lifetime, thanks to macrophages, kind of generations and generations of macrophages living and dying to keep that tattoo in place and shield it away from the body. And that works very well in most cases, but for reasons that we do not understand very well, sometimes these kind of one of these macrophages or many of these macrophages elicit an immune response and call in T cells and call in other immune cells, start an inflammation. And then this thing here that is called sarcoidosis, granuloma formation can happen. And we wanted to understand what's actually going on there. So we took biopsies, or our hematology colleagues, Georg Stari and his team took biopsies, and we performed single-cell RNA-seq and spatial transcriptomics profiling. We get a parts list of all these cells, kind of what these granulomas are consisting of, and we get an initial spatial overview. And then comes the hour of the bioinformaticians, really, to dig deep into these data and try to reconstruct on a biological level what's going on in these structures. And here the result was unexpected. We found macrophages, T cells, and fibroblasts. That was what we were thinking we would find. But those cells, we were looking for disease signatures. We were looking for cells that were malignant in a way, like really aberrant, different from the body's normal immune cells, and that would be the drivers of the disease. But this was not the case here. Those cells were all activated and inflammatory, but they were normal. And there was no reason why they could not go back to a less inflamed stage. So what seems to be, but we saw a very strong crosstalk between the T cells, the macrophages, and the fibroblasts. So what seems to be happening here is that there is a vicious circle of miscommunication where the disease sits not so much in a single cell type that is aberrant, but in a cell communication cycle that is kind of broken and inflammatory. Now, if this is the case, perhaps we can disrupt this. So this motivated our collaborators to conduct one clinical trial for an mTOR inhibitor that was shown in a mouse model to perhaps be able to interfere with at least the macrophage side of the sarcoidosis. And it turns out that indeed dosing for a relatively short-term time with this mTOR inhibitor was able to break this vicious cycle and eradicate these granulomas, these sarcoidosis in at least a significant number of the treated patients. And what is more, the disease did not come back when we stopped treating those patients. Indeed, cancer would have probably come back, but here we broke the link, and then the cells resolved, they went back to normal, and the disease was gone. So this a little bit as an illustration how a single cell and spatial, without having too much of an idea what you're actually looking for, can allow you to go systematically and deeply into human tissues and human diseases. And the Human Cell Atlas tries to kind of systematize this type of approach, make it a really global endeavor based on data sharing, method sharing, joint quality standards, etc., such that over the course of this year, a lot of the kind of version 1 atlases will come out on the website of the Human Cell Atlas. And there's the Human Cell Atlas meeting coming up in Boston in a month and a half or something like that. So if you have time and if you've got interest, I think you can still sign up for that meeting. With all this single cell sequencing, we are creating huge data sets, and that obviously triggers my bioinformatic interest, because many times over we've been confronted with a situation that our collaborators needed bioinformatic support. They had interesting data, they wanted to understand what's going on, and bioinformatics is really in short supply, at least here in Vienna. There's always more interesting data sets that could be analyzed than bioinformaticians that are looking for a project. So what we started at our institute is that every new incoming PhD student generation, they get training to analyze the data themselves. So be them biologists, medical doctors, chemists, etc. We take them all through a crash course in R and Python to bring them to the point that they can look at data with computational methods. And I find this useful, and many students have profited from that. But it feels kind of slightly counterintuitive. It's 2026 now, and we are essentially training kind of really bright students to speak to the computer in a language that was created in the 1980s to be understandable for the pretty dumb computers of the 1980s. So wouldn't it be nowadays, with all the AI around, shouldn't we do it the other way around? Shouldn't we teach the computer to speak our language, the language of our PhD students, let them talk to the data rather than kind of memorize all these complex terms that coding involves? So as a first shot in that direction, we tried to embed kind of a data-aware a single-cell data-aware chat engine into the Cell by Gene browser. So many of you might have worked with the Cell by Gene browser, very useful visualization tool. In the middle, you see a UMap. On the left, you see the tools to kind of navigate and look at this data. And what you see on the right is what we added here as a plugin, Cell Whisperer, a large language model combined with the single-cell RNA-seq foundation model that you can actually talk to your data. How this practically works is you can ask, green is me and blue is the AI, Cell Whisperer AI responding. I can ask about the activity of certain pathways and biological processes, and it will overlay intensity colors here on the left. I can ask follow-up questions. I can draw a circle around cells that interest me in particular. I can ask about what these cells are, what they are doing. I can even ask the computer to speculate about what are the roles of certain biological processes in the cells that I've selected. So really making it the type of dialogue that you might have with a bioinformatics colleague when looking together at a data set. And kind of the key point here is the model Cell Whisperer sees the data, the single cell data, but also has biological knowledge in the sense a large language model would have biological knowledge. So technically speaking, how we do it is pretty similar to how ChatGPT would be able to, when you upload a photo, would be able to tell whether the animal on that photo is a cat or a dog or a tiger or a lion, essentially projecting that picture of the lion into a high-dimensional embedding space and then looking nearby whether there are more kind of text labels and sentences and that refer to lions or to tigers nearby. So practically speaking, what we did is we used standard large language models to curate a massive data set from GEO and Cell by Gene of kind of textually annotated transcriptomes, bulk and pseudobulk transcriptomes. And then we processed the bulk and pseudobulk transcriptomes through a using the GenFormer single cell foundation model, and we took the corresponding text through BioBERT large language model, and then essentially pulled them together using a clip model into kind of a multimodal embedding space, such that a transcriptome in this high-dimensional space is in close proximity to labels, text of labels that refer to similar cell types. And this can now be queried in both directions. We can say, here's my transcriptome, give me a label for that. Or I can say, I'm looking for this type of cell, and I described that cell textually, and it gives me the corresponding transcriptome. And this is essentially at the heart of Cell Whisperer. And then to really make it talk to biologists, we have it learn. We essentially kind of fine-tune a large language model to answer questions about the data in the way that a biologist or a bioinformatician should or would. And this creates this ability to really talk to the model, and you can try it out on the website, the Serviceable website. It works pretty decent for a first shot. If you want to run it with your own data, you have to download the code and download the models, and it's pretty fast to set up, even kind of on your local laptop or medium scale machine. So it's not the training was expensive, but the kind of using it for new data is not very computationally troublesome. And we're trying to go beyond single cell data. We are also looking at spatial information. Pathology, I already mentioned at the beginning that pathology is really the discipline that helped us connect what we are seeing at the tissue level to the diseases we observe in patients. And a pathologist is really trained to see structure in all of these pictures. And we wanted to see if we can take a similar approach to, and like good bioinformaticians, good pathologists are really in short supply, because for many projects you need a good pathologist who would look at your samples. So perhaps the AI can help here. What we did is that we took the H&E stains, this histopathology information, and on that basis we predict with a spatial transcriptome profiles. So essentially predict the transcriptomes that correspond to each of these pictures. And then we use Cell Whisperer to be able to query this. And then you can ask questions like, show me T cells, show me tertiary lymphoid structures, show me blood vessels, and these type of things that a good pathologist would directly be able to show you. I mentioned Karl von Rokitansky at the beginning, the guy who founded pathology as a medical discipline, and his textbook, where both in Europe and the US, kind of the first generation of pathologists were trained on, was a book with no pictures. And I think that's interesting, because pathology, really being the most visual of all medical disciplines, was launched by a book that had not a single picture in it, at least in the first edition. And I think this shows a little bit something fundamental about why large language models work so well. Because if you can describe very well what you are seeing, you can convey the same information that a picture shows in words, and often perhaps even clearer, because you abstract away those things that are less relevant. And while it's totally true that a picture says kind of more than 1,000 words, it's also the other way around. A well-crafted text can really capture the essence of a picture. I think there are a few implications of the current advances in AI for the field of bioinformatics as a whole. First, if you are a bioinformatics tool developer, you should absolutely add an AI-powered checkbox to whatever tool you're developing. And that checkbox should serve three purposes, basically. It should obviously be able to look at the data and help the user talk about the data, interpret the data like I illustrated it for Cell Whisperer, but it should also be able to answer biological questions. So you don't always have to go from your data analysis to kind of Wikipedia or PubMed, etc., to figure out what a gene might be involved in. And obviously, it should also help users to better use your tool and use all the exciting features that you've programmed into your tool. And I think this combination of kind of multimodal embeddings with large language model fine-tuning is a pretty powerful and relatively straightforward approach to build such models. And thinking this approach just a little bit further, if you're talking to your software tool anyway, you're not far from the point where you are essentially supervising bioinformatics agents working for you, where you would essentially tell them the type of analysis you want, and they would query for you databases, process data sets, etc. And I think this has quite some implications for the way we do bioinformatics, because as PIs, as group leaders, we're used to working through the hands of our PhD students and postdocs. We try to think of projects, we try to convey some of our ideas, we ask questions, we tell them analysis that could be interesting, and then they come back to us with results, and we look at the results together. And I think this is, and kind of understand what makes sense and where further analysis is needed. And I think this very much anticipates how the next generation of bioinformatics students will work. Even a first-year PhD student, maybe bachelor master students, they will work like I do. They will oversee bioinformatics assistants working for them, analyzing the data, finding data sets, etc. And that means we need to perhaps dramatically change and improve how we train already in undergraduates in their ability to do research, because then we will not have so much time to slowly acquire the ability to ask good questions and to check plausibility of results. But this is how they will be working from day one, spending much less time coding, much less time kind of on kind of relatively repetitive stuff, but from the beginning have to have the maturity to be able to understand what's the next analysis that they should task their bioinformatics agent to pursue. But there's one thing really that AI as it currently stands is terribly bad at, and that's figuring out causality. And since almost everything that we are ultimately interested in biology is causality, as the basis also for therapeutics, here is really the time to go back to the lab. And often that means after a very systematic single-cell spatial atlas type of profiling, ultimately you select one or two genes and you validate those experimentally. I like to call this one gene, one postdoc paradigm, where you essentially, as a professor, you suggest a gene that a new student or a new postdoc might work on, and then you hope that that works out, and that gives you 4 or 5 years later like a really interesting, really impactful paper. But more often than not, it might not, unless you're pretty conservative with your choices. And I think all the papers on P53 and a handful of very popular genes, this is not only because these are such important genes, but also because it's a little bit of a safe bet when you find one of the usual suspects popping up in your screens, in your single cell data, to do the validation on where you already know something will come out, rather than on like a random zinc finger or SLC protein that might be basically uncharacterized. And here I think the technological advances of the last years are dramatically changing the scene, because we no longer need to make this decision. We don't no longer need to focus on just one gene, but with CRISPR screening and the kind of powerful single cell and spatial readouts, and more complex cellular models such as organoids, even in vivo screens and mouse models, we can now study perturbational causality for kind of hundreds of genes at the same time, even thousands of genes in millions of cells. And this is something that certainly influenced the focus of my lab, that kind of with CropSeq, PerturbSeq, with this concept of combining CRISPR screening, not with a simple cell survival readout, but with something that's as data rich as single cell sequencing, we can do experiments that give us causality at the scale of many hundreds or thousands of genes at the same time. And this technology is becoming broadly useful. And it's not only single cell sequencing, single cell RNA-seq, it's also spatial profiling. We can do CRISPR screens where the readout in the end is kind of a slice of the tumor, and we see where the immune cells are going, or we can use FACS screens, FACS imaging-based screens. So a lot of exciting things to ask causality questions at scale. I should also mention that there is a lot of power in time series. I think we are in the field, we are doing time series not enough, because time series is already giving you something that approaches causality. It's sometimes called Granger causality. It's basically the almost trivial idea that an earlier event can cause a later event, but a later event cannot cause an earlier event. So this doesn't give you direct causality, but it gives you a certain error or a certain level of directionality. And here in essentially a technological demonstration study, we applied this to macrophages doing high resolution epigenome and transcriptome time series, and we integrated this with crop-seq perturbational screens. And this type of approach, in a relatively generic way, provides deep mechanistic functional insight into what's going on, and especially combining this now with more sophisticated models such as organoids, I think a lot can be figured out in a lot faster than if we were solely relying on kind of a single postdoc working out a mechanism truly step by step. I mentioned the human cell atlas earlier. The human cell atlas has huge power in providing a descriptive map of what is there in the human body, in every organ, down to the single cell level, down to the molecular level, but also through spatial transcriptomics data in their relationship to each other. But the human cell atlas, based on tissue, cannot really give causality. So organoids, I think, are filling this gap very nicely, because we can do CRISPR screens in organoids, we can do drug screens in organoids, we can do perturbational screens in organoids. And the great computational challenge is now really the integration of all the single cell data that come from primary samples and the characterization of in vitro models, including organoids, into kind of a Rosetta Stone mapping between in vitro models, which are often faithful for some aspects of in vivo biology, but not for other aspects. And this working out could be a big step towards kind of a more causal understanding of what is going on. Now, in the last couple of slides, I want to describe how we are taking CRISPR screening and computational methods towards cell therapy. Essentially, kind of focusing on this idea that cells can be programmed into something synthetic, something artificial that has very specific and useful biological functions. And perhaps the community has gotten furthest on this with the concept of CAR T cells, the idea that you take immune cells from the body of the patients, and in vitro, outside the body, you essentially target them, you equip them genetically with the ability to recognize cancer cells, and then inject those back into their body, where they go hunting for those cancer cells that they have been kind of genetically trained to pursue. And that works beautifully for certain types of cancers, most notably for B-cell leukemia and lymphoma. But even for those where it works very nicely, CAR T cells often fail. So essentially running out of steam, running out of energy, which is perhaps not surprising, because the cancer microenvironment is a really tough place to be in. And often the T cells that these CAR T cells are made of, they are already from heavily pretreated patients. So we asked whether we could use some of our screening technologies to fill in for this gap in evolutionary optimization, that CAR T cells have never been made by nature to do what they are doing, but we are reprogramming T cells that have a lot of other functions as well into something that's kind of single-minded tumor cell killer. And our idea is that perhaps CRISPR screens can provide a form of artificial evolution to make these CAR T cells adapted to their new role and become more effective killers of cancer cells. To be able to do that at scale, we built an experimental platform for CAR T cell screening in vitro and in vivo that supports CRISPR knockout, base editing, combinatorial screens, also multiple readouts. Cell fitness is a good readout, but FACS screens and single cell sequencing can provide a lot more information. And all of this needs to be scalable to not only test kind of a handful of genes, but really go genome-wide with these screens. And genome-wide we went with these screens initially in vitro because in vivo screens are really technically not feasible to do with good coverage on a genome-wide scale. So we wanted to initially cast the net wide, and we defined interesting biology of CAR T cells, such as the ability to engage the tumor cells, their ability to activate, their resistance towards T cell exhaustion, their resistance towards apoptosis. And we did many of these in vitro, several dozens of these in vitro genome-wide screens, and all of those gave us genes, interesting genes, some well-known genes like PD-1 and CTLA-4, genes that we know well that TIGIT, genes that we know well have important roles in biology, but also many genes, or in immunology, I should say, but many genes also that were new and interesting. And based on this, we came up with a list of genes that we were excited about from many of these in vitro screens. But when we started testing just some of these, it was clear that by no means all of those would validate in vivo. And so we needed a second type of screen, a screen in mouse models that would sort out the complexity and where the in vitro situation is quite different from the in vivo situation. In vivo CRISPR screens are still methodologically non-trivial, and we made some improvements to the setup. And with this setup, we were then able to test all our 39 genes that we ranked very highly and saw which ones that were really helping our patients. And then we validated, and then we had a 100% success rate validating our hits. We found some genes that already quite well understood in the context of CAR T cells, such as FAS and apoptosis mediator. But we also found, and this is why I like screens so much, something that you would never have expected based on prior biological knowledge. We found raw G here and extensively validated that raw G knockout CAR T cells were substantially better at clearing the tumor than standard wild-type CAR T cells. Now, raw G it's an absolutely non-obvious choice. Raw G is a signaling protein that has many roles in immune cells, in actin cytoskeleton regulation, immune synapse formation, T cell activation, but it has been found in patients to be, when abrogated, to cause an immunodeficiency. So those patients where this gene is gone actually have weaker immunity, while we want to make the CAR T cells stronger. So we couldn't believe it initially. We did a lot of follow-up. We found that these CAR T cells were, with raw G knockout, they were proliferating harder. They were better in kind of acquiring a central memory phenotype, and they were less likely to exhaust. And we looked deeply, and essentially our understanding as it stands is that the CAR T cells that we remove raw G from, these are specialists. They are really good at going in and hunting the tumor cells, whereas normal T cells, and this is what CAR T cells usually inherit from the T cells, they are generalists. They're not so specialized, and this is why they're not as good as hunting tumor cells. So this is exactly what I meant earlier when I said in vitro evolution, we are, or artificial evolution, we are making CAR T cells here specific for the task that we want them to be used for. And we are quite excited taking this into phase 1 clinical trials. But obviously that comes with its own challenges. We decided that it would be very useful to switch to a base editing setup instead of the standard that CRISPR knockout to avoid the genotoxicity of DNA double-strand breaks. And we have to adapt our workflows to GMP-compatible manufacturing. Fortunately, we have very good clinical colleagues here in Vienna that we are working towards the goal of testing RG knockout CAR T cells in patients. And then we hope that these CRISPR-boosted CAR T cells will perform as well in patients as they do in mice. Finally, the big challenge for CAR T cell therapy at the moment is the very high cost and effort of making them in a highly personalized, made-to-order way for every new patient, because you take the blood from the patients, you engineer those cells, you give them back to the patient. And an exciting approach to overcome this would be in vivo engineering, where you're essentially giving the CAR, the cell therapy, as a gene therapy, such that the vector itself finds its target cells and makes CAR T cells in vivo. Now, this is very exciting, but a big issue is specificity, because you don't want to make all kinds of cells in the body expressing this antigen receptor that is characteristic for the CAR. In particular, you don't want to have regulatory T cells express it, because that could perhaps even tone down the immune response. So one approach that my lab has recently gotten really excited about is AI-based enhancer design. So perhaps we can, if we cannot deliver the virus specific or the lentivirus with the CAR specifically to the T cell subset that we are interested in, maybe we can program a promoter enhancer construct that will ensure transcription of the CAR only in the cell types where we intended to do. So we've built the DNA craft methodology, regulatory elements by conditional Monte Carlo tree diffusion, essentially a diffusion model, where we want to design enhancers that maximize the cell type specificity between desired and non-desired cells. So you really give the AI model a positive list and a negative list. I want my gene expressed in those cells and give them the single cell multi-omics profile of those cells, but not in those cells. And then it will find a way to optimize the DNA sequence of this enhancer and promoter location to be as discriminatory as possible between the different cell types, while also adhering to the general grammar of the human body. And how we do this practically is we train a diffusion model that knows how enhancers in the human genome look like. So if you give it a masked DNA sequence that should be an enhancer, it can bring it back to a typical enhancer in a given cell type. And we combine this approach with Monte Carlo tree search, essentially kind of hierarchically traversing a tree and slowly unmasking the sequence that we want to design in a step-by-step manner, and always scoring how well it acts as an enhancer in the positive list of desired cells, as opposed to the negative list of undesired cells. And so step by step, we can build many different versions of enhancers that are highly cell type specific that we can then test experimentally. And we've validated this approach based on single cell ATAC-seq activity in many different immune cells. And we found that our model is indeed able to find highly specific enhancers for many of these settings. Still struggles a little bit distinguishing very similar cell types like CD4 and CD8 cells. But this can also be fine-tuned with additional data in a kind of a lab in the loop type of setting. So the next step really is to experimentally test some of these predictions and to feed that data back into the model, make even better models, and eventually test the performance of CAR T cells engineered to express the CAR specifically in effector cells and memory T cells or in Tregs. So to sum up, I'm really excited these days about combining single-cell and spatial profiling in diseases, in large atlases, with AI models that can kind of learn the biology that's going on, and then predict with the help of AI how we can reprogram cells, essentially getting into a read, learn, write, repeat type of circle of iteratively improving cells as probes and therapeutics. And I would argue that by building cells, programming cells, we will also learn a lot about true functionality that is relevant for kind of human in vivo biology. And with that, I would like to conclude and thank everyone who has contributed to this work.
All right, thank you, Christoph. That was fantastic. It's really, really exciting work. I don't know if I think if people have questions, please put them into the webinar chat and we can answer them as we go along. But I have a few questions. I don't know if you have questions, Liang.
Yeah, maybe you can go first.
Yeah, I had a quick question about the Rho-G. That's really, really exciting. So you mentioned I mean, they basically become super killers, but do you see improved longevity of the CAR T cells as well?
We see a higher memory phenotype, and yes, we also see improved longevity and also better behavior in challenge research, re-challenge experiments. But I wouldn't really call them super killers. It's more that normal T cells have like a attention deficit
Yeah.
hyperactivity phenotype. They want to run around the body, and it's probably useful because the CAR Ts, there's only very few memory T cells really for one specific antigen. So they need to be mobile and then they need to check out many different places. And now in a CAR T cell therapy setting, you flood the body with millions and millions and millions of CAR T cells. And if they all run around, they see so much antigen, they will exhaust pretty quickly. So they should kind of just stop running around and focus on the one thing they're there for and rely on kind of the neighbors to take care of and kind of kill the next cell that's there. This is a little bit of an overinterpretation of our data, but I think this is kind of my understanding. When you do synthetic biology, when we really program cells to do new things, we need to make sure that we fill in for the lack of evolutionary optimization and train them to do the new thing well.
Yeah. Yeah, I think that's a beautiful demonstration of that approach. Yeah, I had another question regarding the last little bit about DNA craft. I could see that being extremely useful very broadly. Are you looking outside of the immune system at other cell types, developing specific enhancers for expression, targeting different subtypes?
We are very excited about the methodology. We see the methodology as general and broadly applicable. The bottleneck is experimental testing because at the moment you still need to generate a few dozen enhancers predicted, get those synthesized, and then test them, and one or two or three of those will work. So it's not something that can fully replace biology at the moment, but I think the potential is huge. You can, especially if you think also reporter constructs, you can make reporters that combine several pathways that sense complex phenotypes And then you can FACS sort those cell types, and you can do CRISPR screens for those with the FACS readout. And a lot of power is in kind of made to order AI-based enhancers, not only for therapeutics, as I illustrated today, kind of targeting gene therapies to where they are needed, but as research tools. And I think there we are totally there already. We can generate these enhancers pretty rapidly. The bottleneck is the experimental testing, but then with the right biological collaborators, that's kind of hopefully something we can fill pretty rapidly.
Yeah, that's really exciting. From both angles, absolutely. I could see how that'd be applicable to our organoid models, our skin and inner ear models. So I might follow up about that point. Liang, did you have a
Yeah.
question?
Yeah. Well, thank you so much for the great presentation. It's very impressive work. I actually, out of curiosity, right? So I think you actually published like Cell Whisper, and also now I'm actually also excited to hear that you actually have Spot Whisper. It mainly support spatial transcriptomics data. I was just wondering what type of data is actually supported in the Spot Whisper. Based on the name, it looks like this is spot based, right? So like 10x Visium is spot level, resolution. I think we right now we have subcellular levels, spatial transcriptomics data like Vism HD, and then some like imaging based data like Murphy, and other type of data. So can you talk a little bit more about the Spot Whisper tools? What type of data and then what type of analysis we can do using your tool?
So Spot Whisperer works such that the input is a histopathology image, and then it infers computationally Visium-like data, and then you can query the Visium data with AI. Now, rightly so, you will say that the old school Visium spot-based approach is slightly kind of last year or the year before, and there is much cooler technologies around now. And yes, we are building a new version that is kind of better and more exciting. This is work led by Moritz Schäfer, the first author of the Cell Whisperer paper. He's now at Stanford in a kind of between a clinic and an AI lab, where he wants to build like a truly multimodal spatial transcriptomics plus text plus image model. There is just one. There will be a paper about that on the upcoming ICML AI conference, and I think that's pretty exciting. So this is on the way, but I don't think it's on archive yet as a preprint, but should come out soon.
Great.
Nice.
Thanks. Yeah, I think just want to remind everyone, like if you have question, type in the Q&A session. You can also raise your hand. We can actually unmute you.
So there was one question in the chat which I can answer.
Yeah, and that was my question as well.
Okay.
It's a good one. Do you see it?
Yeah, can end users swap the underlying foundation model of Cell Whisper, for example, from GenFormat to SCGPT or any? Yes, absolutely. We have tested several models for the paper, and there it didn't make much of a difference. But these models are all quite old by now, so kind of the new generation of models that are trained on much more data, also perturbational data. This is super exciting, and we made Cell Whisperer quite kind of modular about the foundation models that are underlying it. So what you can find on the Cell Whisperer website is obviously you can try out the tool. That's interesting, but the two things that are even more useful than just playing around with the tool, especially if you're a computational biologist, is the code and models. So with that, you can rapidly set up your own version that you can then fine tune to your organ. So if you're studying, you have kind of large proprietary data sets for the liver or the lung, etc. You can really fine tune this to become a liver or lung or organoid model. And secondly, we can, so it is quite modular in terms of swapping the the different models. And what we also make available on the website is this 1 million annotated transcriptomes data, bulk and pseudo-bulk, with indeed we burned more on this, a lot more GPU hours for CellWhisper on the curation of the data sets. So if you're looking for a well-curated transcriptomes of everything in GEO and cell by gene, this is the most curated data set that you will get out there. So if you want to train your own models, you think that our kind of the models we've been using are a little bit too old already by now, then kind of at least download our data set, because that is not going to get outdated very quickly.
Nice. Thank you. We've got another question here from Yuming, which is interesting. They're wondering how AI hallucinations are controlled or resolved or guardrailed in Cell Whisperer and Spot Whisperer.
Yeah, that's my favorite question. Yes, CellWhisper hallucinates, but so do my bioinformatics postdocs. They also hallucinate. You look at data and you think, oh, I've heard that name of a gene before, and I think it's involved in breast cancer. And then you look it up and it was actually ovarian cancer. That's a form of hallucination, but often, and most of the time, that's the end of the story. But sometimes an idea is born out of that. You read up a little bit more and you understand that perhaps there is an overlooked role in ovarian cancer. So we see CellWhisper in its current form as an exploratory tool, and an exploratory tool is okay to have a healthy level of hallucination, especially if it's productive hallucination in the sense of plausible things, because sometimes they might be useful. We could probably train all hallucination or most or all hallucination away from CellWhisperer by kind of very harsh fine-tuning or kind of user in the loop feedback, but we will probably make it boring. Then it will answer like a lawyer, being super formal, super standardized, super safe. And in the exploratory data analysis, I don't want something super reliable. I want something that gives me ideas. And then obviously that comes with a health warning that whatever an AI system kind of spits out needs to be validated further. And we are kind of something that's not quite ready yet to be shown, but part of it you will already find in on GitHub from us is Mr. Biomix, which is essentially a kind of engine for validating results on real data. So the idea is that eventually the AI will call in an agent to verify with kind of classical statistical methods what it has just claimed based on public data that it finds. And I think that is necessary. At the moment, this is still the biologists or bioinformaticians who have to do this, but eventually, this is what I said with we are going into kind of the agentic age of bioinformatics. This is something that a good AI would eventually do themselves.
An excellent answer. I tend to agree about that. It's a good starting point for further investigation. I don't know, Liang, did you have any other questions?
I'm okay. Thank you so much. I
Yeah.
think there's another
I,
question from
yes.
Oh,
Yushin. Yeah.
another question here? Oh, yeah.
Raise your hand.
Yes, yes. Hi, Dr. Bock. Thanks for a nice talk. I do have another question related to Cell Whisper, especially for the part of training based on the conversation or chat and answers. I found it pretty interesting that the training for this part actually also use like AI generated answers. It seems based on like the supplementary notes, you did create some questions, but then for the answers, you also use some other models with the input of dataset metadata, top gene set, and also expressed genes. Then the answers were actually also generated by the AI, different models for sure. Then you train on these questions and answers like again. So I'm just wondering, do you see any like limitations to this approach? For example, if I ask some other questions, like not in your training scope, And also because the answer generated only take into consideration of those top genes, then how well does it perform if I ask some question related to those either less represented genes or the ones that are not in your answer set. So yeah, basically that's my question.
Wow, that's a lot of complicated questions. So first of all, just for the record, I do not recommend training AI solely on AI. That is a very slippery slope that goes bad relatively rapidly and sometimes without you realizing that that's happening. But we had kind of one ground truth point here that what kind of always the link between the text and the genes was kind of created through actual data. So we always took a gene expression profile and its textual annotations. And then the textual annotations were used, kind of further processed with AI, but kind of the link between transcriptome and text, that was always for real. And we also generated simpler questions where we just asked about kind of whether a gene is active and but kind of adding these more complex questions makes it more realistic and kind of safely doing this. AI generated questions and answers can help avoid your second problem, or at least you reduce the problem of the issue of the second problem. So if you fine-tune your large language model, the chat engine, only on relatively simple questions and answers that you can generate in a rule-based manner, then it will get quickly out of control when you ask more complex questions. So the chat allowed us to ask, AI-generated chats allowed us to train the model for more complex questions. Still, we did not go all the way. Like you can really confuse the model if you ask the model like what day is today, and then it will tell you something about genes. So it does not allow you can. It's easily like there's no protection against jailbreaks in there. So if you ask it stupid questions, you will get stupid answers. And that's acceptable because this is a science tool where you can trust that the users are responsible human beings, and it cannot really destroy anything. It cannot give you anything that's dangerous or whatever. But yes, that is a warning that if you operate a Salvador outside the scope that it's trained on, it will probably start giving you less and less reliable answers.
Yeah, I see. Yeah, definitely. Thanks for the answer. Then I guess the current service for doesn't work on like perturbation effects, but do you have plans working toward that direction?
I think that's a very exciting direction. And yes, adding the ability to predict perturbations would be extremely powerful, but it's also hard. Like you've probably seen a couple of benchmarking studies now coming out that basically say it's hard to beat a linear model on perturbation prediction, even with the most sophisticated AI models. I think that's to a significant degree not a problem of the models, but a problem of the data. And this is being addressed with crop-seq, perturb-seq data sets that are really done to scale. So I would think that this problem will disappear eventually, and that will open the perspective really for a version of Cell Whisperer that can anticipate perturbations.
Thank you.
Okay. If there are no more questions, I think we're at time, right, Liang?
Yeah, right timing.
All
Yeah.
right. All right. Thank you again, Christoph. That was wonderful, and I'll be in touch. It was really exciting work.
Sounds great. Thank you very much.
Yeah.
Thank you both.
All right.
Have a nice day.
You.
Bye.
Bye.
See ya.